Hello everybody, welcome to a new Revive webinar by the Global Antibiotic Research and Development Partnership, GARD-P. My name is Astrid Pence-Moore and I'm hosting today's session, Clinical Development for Non-Developers Part 3, Non-Traditional Approaches. For those who don't know Revive yet, Revive is GARD-P's education and outreach program, and it aims to connect and support the antimicrobial R&D community by facilitating learning, sharing knowledge, and connecting people. This webinar series is part of our educational activities. All webinars are recorded in full and can be viewed after the live broadcast on our website, revive.gardp.org/webinars. There you can also find part 1 and 2 of this series, as well as all other recordings. Today we will have 4 presentations, which will be followed by a Q&A session. You can submit your questions at any time during the webinar via the questions window in your webinar control panel as shown on the slide. We will do our best to respond to as many questions as possible. Now I have the pleasure to welcome our speakers. David Shlaes, who already spoke on parts 1 and 2 of this series and who will also moderate today's Q&A. And Ian Friedland, a clinical consultant, Sumathi Nambiar from the FDA, and Mair Powell from the Irish Health Products Regulatory Authority. And with this, I'm handing over to you, David.
Okay, thank you very much, Astrid. I'd also like to express my thanks to GARD-P for their continuing work in trying to provide educational opportunities for the world of antimicrobial products and antimicrobial product development. Let's see if I can get to the first slide. There we go. Oops. Okay. So I guess, first of all, I should say something about the fact that this is a delayed webinar. I'm sorry that it was delayed. It was originally planned to take place in January, but because of the US government shutdown, and because we felt that it was very important to have all of our participants actually participate, we moved it to this date in March. So I apologize for that. The topic that we're going to talk about today is probably the most difficult of the topics we've covered in our 3 webinars, because as the title suggests, because these are non-traditional approaches, it means that none of us have a lot of experience in developing drugs along these lines. And there are a lot of questions about how to do this, both among people who are experts in the area and people who are not. So today we've brought together, once again, people, not me, but people who are actually on the front lines of this question, dealing with companies and others who are trying to develop these approaches in a real effort to try and bring innovative products, innovative strategies and products that will help us in the fight against bacterial resistance to antibiotics. So I'm very glad that we have the quality of participants that we have. So we can't cover everything today. We'll try and stay with systemic therapy, sometimes prophylaxis, of serious infections due to more standard pathogens, not mycobacteria, for example. If you want to know about traditional small molecule so-called enhancers, which are combined in a fixed dose with a known antibacterial, then you should go back to webinar 1, which is available on the GARD-P website, Revive. We may briefly address certain non-traditional goals of treatment, such as prevention of resistance and how does one demonstrate a real clinical value there, or prevention of superinfection, such as C. difficile, perhaps looking at protection of the microbiome. Some approaches use proteins, peptides, or hybrid molecules instead of small molecules. These could be antibodies designed to inhibit bacterial growth either directly or indirectly. These could be licensed. There are a number of licenses that are currently in various stages of development. If used for standalone therapy, not prophylaxis, but therapy, the clinical trials to support these products will resemble those for traditional small molecule antibacterials. And again, you should go back to webinars 1 and 2. Other aspects, though, of development, such as safety and CMC, may be regulated quite differently than for small molecules. So non-traditional approaches might include inhibitors of virulence, for example, and there are a number of projects seeking to develop these approaches. Examples would include type 1 secretion inhibitors or quorum sensing inhibitors, and there are a number of others. They might be looking at inhibitors of non-essential bacterial functions, such as, for example, inhibitors of REC-A. They might be looking at immunomodulators, looking at attempts to alter host functions. And any of these could either be small molecules or proteins, but all certainly would be non-traditional approaches to antibacterial therapy. When you're thinking about this and you're thinking about actually bringing this to the marketplace and to clinical utility, you're going to have to know how to dose patients. If you can carry out pharmacokinetic, pharmacodynamic analysis in preclinical models or in the clinic, that's going to be a very important way forward. But this may not be possible for a number of these approaches, in which case you're going to have to think about actual dose ranging studies in patients in phase 2 or phase 3 clinical trials. You could consider developing some of these as fixed dose combinations with a traditional antibacterial, where a more standard approach, non-inferiority trials, can be undertaken. But there is a hook here, and that is that you still will have to develop, you'll have to demonstrate that there's actually a value in the to the new therapy in the clinic. If you're developing the therapy, the non-traditional therapy, as an adjunct to standard of care, this is going to require superiority trials. And this is another area of great difficulty, I think. Now, bacteriophages is obviously another approach that people are taking. Again, if it's a standalone therapy of serious infections and you can gather enough patients, clinical trials may resemble those for traditional antibacterials. But since these are living organisms, the regulatory approach to CMC and safety is quite different than for small molecules. Now, as I'm trying to point out, these approaches are not without risk. So again, if a molecule has no or minimal intrinsic antibacterial activity and cannot be used as monotherapy, and this would include the virulence inhibitors or inhibitors of non-essential bacterial functions, immunomodulators, and other approaches, the effect of the molecule on standard therapy must be a dramatic one, and I'm going to give you some numbers around that in a little bit. And in this case, this dramatic effect must be visible in vivo. In addition to this, and I'm going to get into this a little bit again later, you have to demonstrate the product's clinical value, not just for regulators, but for patients and physicians, payers, and everybody else who needs to understand how these molecules can be used and what their advantages are. So these, of all of the topics that we've discussed in the previous webinars, these may be the most difficult and certainly the highest risk of the examples that we have discussed and we are discussing. If the product is going to be specific to rare pathogens, which is likely, especially for the virulence inhibitors, for example, there is again going to be the problem of trial feasibility that we discussed back in webinar 2. Not that you can't overcome this, but this is something that you need to keep in mind. And then for many of these, there's the additional challenge of the fact that you have to change the way physicians normally use antibiotics and their reliance on susceptibility testing. It requires kind of a paradigm change for the patients and physicians who will be using the products. Again, I don't think it's insurmountable, but it is a challenge. So I want to say something about superiority trials, because again, if you're going to develop your new therapy as an adjunctive therapy, so an add-on to standard of care or add-on to so-called best available therapies, the trial design is going to be important. And this table comes from an IDSA publication from back in 2012, looking at superiority trials in infectious disease. But what I want to point out is that first place, to identify infections where you could theoretically show superiority, you're going to have an endpoint where ordinary antibiotics, best available therapy, has a response rate, say, in the 50 to 70% range. And then you're going to have to show that your therapy approaches 20% better to get down to any sort of reasonable trial size, assuming that you want to show this difference at a p-value of 0.05, which maybe is not always necessary. But the advantage of superiority trials is that you show the value of the product that you're developing. One of the disadvantages, and another one of the advantages, by the way, is that the endpoints do not have to necessarily be the same endpoints that have already been defined for non-inferiority trials of antibiotics. The disadvantage is that antibiotics basically work quite well. And the kinds of patients that you're going to be able to recruit into your trials where they don't work well are going to be those that are the most sick and where you have the greatest risk of either one, not being able to enroll your trial, or two, not being able to show the kind of superiority that you'll need to show. So just keep this in mind. Now I want to get back to this idea of the value proposition and regulatory requirements. These are obviously overlapping concepts. The regulators are going to want to see that the add-on therapy or the additional therapy that you're proposing, if it's an additional therapy, or if it's a standalone, they're going to want to see that it has value, clinical value. But it's not the same as the considerations that patients, clinicians, payers, and investors might want to see. So it's one thing to get regulatory approval, and it's another thing to get acceptance in the marketplace. And these two are I think, different and worth exploring further, and we will as we get into the question session, I think. So a few thoughts for those of you out there who are in companies. I would say, first of all, have a portfolio, and in your portfolio, balance risks. I know of at least 2 companies that started out working purely on virulence inhibitors. Both of those companies now have their most advanced products are actually more traditional antimicrobials. So balance your risks. When you start, as we talked about in webinar 1, have a target product profile and develop a value proposition upon which this target product profile is based, and have an idea of how you're going to fulfill your value proposition as your product goes through development. For peptides, proteins, bacteriophage, antibodies, and other less common approaches, you need to seek early advice from regulatory agencies on CMC and safety for sure. And for any therapy that will be adjunctive, or any therapy where the population to be studied is small, such as in some of the pathogen-specific indications we discussed previously, you need to be seeking early advice from regulatory agencies on the design of the efficacy trials that you would pursue. Okay, and with that, I'm going to ask Dr. Nambiar to step up. Dr. Nambiar, as you know, is Director of the Division of Anti-Infective Products at the FDA. So, Sumathi?
Yes, thanks, David. Thank you all for joining us at today's webinar. Okay, so what I'll try to do in the next 10 minutes or so is provide some clarity on product definitions and jurisdiction, discuss some important considerations in non-traditional product development, talk about a couple of lessons learned. We don't have a lot of experience in this particular category of products, as David has already mentioned, and then just summarize some highlights from a recent public workshop. So now there's a lot of confusion regarding what is considered a traditional therapy and what's considered non-traditional therapy. So it really depends on how you want to look at the product. If one considers all small molecules to be traditional therapies, then non-small molecules such as monoclonal antibodies could be considered non-traditional. Another way potentially of looking at it is based on how it acts. If a product has direct antibacterial activity, one could consider it as a traditional therapy, whereas products that have an indirect mechanism of action, such as an antivirulence factor or host-directed therapy, one could consider that as non-traditional. So rather than focus on nomenclature, I think it's more important to consider what is it that the product can offer, because that really will impact on the types of studies and the development program. I know David has already touched upon this, but a very important question to consider is what benefit will the product provide to patients? So what is the clinical benefit that one expects this product to provide? Because, you know, one can do a lot of work in the non-clinical space, one can delineate and try to understand what the mechanism of action is, but at the end of the day, it has to have a meaningful impact and meaningful clinical benefit to patients. So that's a very important question to consider. And how can one measure such a treatment effect? Then in terms of what the development program would look like, I think what's very important is if the product has to be used alone or whether it can be used only in combination with standard of care. Then whether there are specific safety considerations for the product, both from a non-clinical and clinical standpoint. And then also, are there any specific pharmacokinetic or manufacturing considerations? So it's important to ask oneself all these questions rather than just to get too worried about whether it's what category of non-traditional therapy it necessarily would be. You know, we've seen some products come through in development. We don't have a lot of experience, but this is an example of list of some of the types of products we've seen. A fair number of monoclonal antibodies are in different stages of development. They're either targeted to treat an infection or in some instances to prevent an infection. We've seen some examples of host-directed therapies, gut microbiome directed therapies, phage products such as license, and others that I've listed here. Our regulatory definition for biologic products, I've provided a website for you, which goes into great detail about what's considered a biologic. I think certain biologic products are under the purview of the Center for Biologic Evaluation and Research, CBER, and some others are under purview of the Center for Drug Evaluation Research, CDER, where I work. And in the next slide, I'll give you some examples of that. So products such as monoclonal antibodies, phage products such as license, immunomodulators are under CDER's purview, whereas products such as vaccines, phage therapy, blood components, etc. fall under CBER's purview. In the anti-infective space, we have not had a lot of experience with biologic licensing applications, but we have 3 products, 3 approved BLAs. The first 2 are approved for the treatment and prevention of inhalation anthrax, where the efficacy was demonstrated in animal models of infection, and besletoxumab is a monoclonal antibody that was approved to reduce the recurrence of C. diff infections in a certain group of patients. So some important considerations in developing such products from a non-clinical standpoint. I think we have to have discussions around the appropriate species selection, what the potential for immunogenicity of the product would be. And in many instances, there is a need to modify the battery of non-clinical studies. It may be a little different than what one does for traditional products. From a clinical pharmacology standpoint, we are used to using MIC-based PKPD approaches for traditional antibacterials, and they may not necessarily be applicable to some of these products. There might be a need to evaluate mechanism-based PD effects, such as neutralizing antibody, and sometimes if there are any clinically relevant downstream effects because of the immunological nature of the product. From a clinical standpoint, I think generally speaking, the approaches to develop these products are in great part very similar to that of a small molecule drug development program. There might be some subtle differences. And I think David has already mentioned this in his presentation. I think if the product can be administered as monotherapy, then one can choose to do either a superiority or non-inferiority trial. It'll really depend on the clinical condition being studied, what endpoints are being pursued. But for products that are added on or given as adjunctive to standard of care, then superiority trials will need to be conducted to demonstrate the added benefit of this adjunctive therapy over standard of care. If one is considering using novel endpoints for which we don't have experience from prior trials, I think you have to take a lot of time and give it a lot of thought as well to ensure that the endpoints being proposed or the endpoints being studied are clinically meaningful, and certainly would be helpful if these are evaluated in phase 2 trials so that one can make a meaningful decision in moving into phase 3 and choosing the most appropriate endpoint. Safety considerations from a clinical standpoint, will really depend on the nature and intensity of the effect on the specific target and non-targets. And also with foreign proteins, one always has to keep in mind the potential for hypersensitivity reactions. So as I said at the outset, we don't have a lot of experience with these kinds of products, but I just have 2 examples that I thought might be worthwhile discussing. In the antibacterial space or the traditional antibacterials, there are many important lessons that we have learned from clinical development programs where sometimes it's a translation from preclinical to clinical that has not worked out well, or we've had issues with dose selection, etc. So I think it's always important to keep in mind that there are many things that clinical trials will teach us, and it's very important to have adequate, well-controlled trials for us to be able to assess the benefit of these products. So bezlotoxumab, and again, all of this was discussed at the workshop and also at an advisory committee meeting. So it's in the public domain, and I've given you the link here. So early in product development, the hypothesis here was that the antitoxin A antibody or the combination of the 2 antibodies would be needed. And then the first phase 2 trial was evaluated because there's emerging non-clinical data that suggested that in fact the combination might be better. And then the next phase 2 trial was done where the combination was studied. It showed that potentially there was a treatment benefit here. And then the phase 3 trial design had to be factorial because one had to demonstrate the contribution of the components. And interestingly, this study showed that the antitoxin A antibody alone was in fact associated with poor outcomes. And it was the antitoxin B alone that was effective, and the efficacy was demonstrated with the antitoxin B alone. And that's the approved product and not the combination. I think many of you must be aware of this press release from Arsenis on their anti-staph antibody, where the phase 2 trial had to be terminated early for futility, and this was an anti-staph antibody for preventing Staph aureus pneumonia. We've had a series of public meetings, not all of them necessarily talking about non-traditional therapies, but there's relevant information here which might be of interest to those of you developing such products. I'll just touch briefly upon the last one, which was an FDA workshop, which we had in summer of last year. At this workshop, we presented, non-clinical, clinical pharmacology, and microbiology considerations for non-traditional therapies. I think All this information is available on our website. We discussed 4 hypothetical cases. One was a monoclonal antibody for prevention of VAP infection. I don't remember right now whether it was Pseudomonas or staph. Then the second one was to reduce the risk of infection due to an MBL-producing organism by preventing acquisition of the same. We had discussion around a gut microbiome-directed therapy, and the last one was a bacteriophage endolysin. And we received, in addition to the panel discussion, we had presentations by not just the regulators, but also academia and industry. And all of this information is available for your review. So I think before I close, I just wanted to emphasize, and I think David had made this point as well, given that we are all learning and there is, the development of these products and we don't have a lot of experience, it's very important that you engage in discussions with the division early on, and we're certainly willing to interact and have a dialogue with you as often as needed. And the development programs, it's probably not going to be a one-size-fits-all approach. There would be differences depending on the nature of the product. We highly recommend that you use the pre-IND consultation process that's available, and also to keep in mind that the CMC aspects of the program are very important, and I've provided you the contact information for the division. So in summary, I've outlined some regulatory considerations for the development of non-traditional products. I think it's fair to say in general, there are many similarities with the development of small molecule drug products, but there might be some unique considerations that are very specific to the product being developed. Clinical trial designs will depend on the expected treatment effect and whether the product will be used as a standalone or will be administered as an adjunctive to standard of care. And once again, I highly recommend early interactions with the division so that you have a successful development program moving forward. I've given you a couple of slides of references which might be useful, and that concludes my presentation. Thank you very much. Turn it over to Mair. David, would you take over?
Okay, yeah, next up is Mair. Let's see who has the slides. Yeah, so our next speaker is Dr. Mair Powell from HPRA in Ireland, but of course she's been a very important person in the EMA approach to infectious disease. So I'll turn it over to Mair.
Yeah, good afternoon, everyone. So indeed, as David has said, I've now moved to the Irish agency, but in fact, I'm talking to you all from London because I pretty much work at home these days. So I'm going to be underlining quite a lot of what you've already heard, so I can probably go through this quite quickly. But nevertheless, it's probably good to hear that people are thinking generally along the same lines. So indeed, I agree with Sumathi that one can fret about the classification of various agents, but we tend to look at it rather simply. Generally, we're looking at these family of hugely variable types of interventions, generally as those that are not specifically within the scope of our revised antibacterial guideline. So they may not be acting as standard direct antibacterial agents, or they may be doing things by completely different ways. And some of the things that we have been approached about so far are listed here, but indeed there are others. And the microbiome modifying products for some reason seem to be extremely popular in the EU. We've had quite a lot of interactions with companies developing alternatives to raw fecal transplants, for example. And indeed, as others have said already, we're looking at these depending on whether they're being used alone for treatment or prophylaxis, whether they are adjunctives to standard antibacterials, or in some cases, rather than being adjunctive, are being proposed to us as sequential to standard antibacterial agents. We have recently released a revised guideline on antibacterial agents, which is out for consultation until the end of July. Now, as I've said, specifically, the scope says that the sorts of things on my previous slide is not really included. Nevertheless, in all instances, I would agree with Sumathi that the general principles apply. And although one needs to tailor to each product, I think pretty much 90% of what we say in that guideline is of importance and relevant to the types of products under discussion today. As others have said, a demonstration of a clinical benefit will be required or must be predicted from biomarkers with a very high degree of confidence. I don't know that we've got there yet, but we wanted to at least lay open that possibility. And the program indeed will depend, as I'll talk in a moment, about whether it's going to be used alone or not. Importantly, each product will need specific consideration. There really is no one size fits all here. And I would recommend that CHMP, that is centralized EU advice, is obtained, and probably on several occasions. Just to remind everybody how it works in the EU, which is rather different to the US because we have to deal with many member states, companies can choose to go talk to individual national regulatory agencies, and some of them like to do that first before they come for CHMP scientific advice, which is coordinated by EMA. Others like to do both, and others, perhaps particularly the smaller companies, generally tend to go straight to CHMP. This advice can cover any aspect of quality, whether it's biological or non-biological in terms of the product, non-clinical, clinical development, and in one or more separate or combined procedures. So questions on all three parts of the dossier can be put into one advice if that's what people need. Face-to-face meetings are generally held with national regulatory authorities. We don't always have a face-to-face meeting during CHMP advice. We tend to particularly do that when we need to ask the company additional questions before we can reasonably write good answers to their questions to us. So we try to have as many face-to-face meetings as possible, but the numbers we see are so large that we have to be a bit choosy. Written answers are then given from CHMP and indeed from some national regulatory agencies. Rather than just commenting on minutes, some agencies actually write formal answers, which I think can be very helpful for companies, and they can then use them to submit to other agencies. It's especially important to discuss the clinical programs with the CHMP, and particularly in these instances where the written guidance is not specific to them or indeed experience is lacking. Now, Sumathi touched on non-clinical, and indeed in the EU we see probably that proof of concept for many of these products is going to come initially from in vitro and non-clinical models. There may also be clinical trials in the target population that could be viewed as proof of concept. For each type of product, depending on the molecule, there's going to be a need to consider what has to be done from the clinical pharmacology program standpoint. Others have said that the PK/PD relationship, we may not be able to get very much for certain types of molecules from attempts to establish PK/PD relationships and analyses. But it is important for some of these molecules to understand the potential for any off-target pharmacodynamic effects. In some cases, we feel that the only way to select a safe and potentially efficacious dose may be from clinical dose finding, something that is almost out of fashion now with standard antibacterials. But for these types of products, if it's not going to be possible to establish a PK/PD analysis or a robust analysis, then probably that's what it's going to come to. And these, of course, can be very difficult. The pivotal trials, as others have said, we do need a clearly defined primary endpoint, and that has to be pre-agreed with us on a case-by-case basis. For adjunctive use, indeed, I agree that we're going to be looking for superiority for the adjunctive treatment plus standard of care versus placebo plus standard of care Generally, where standard of care will include a standard antibacterial therapy. On occasion, though, maybe the adjunct is going to be given in a situation where there is no licensed standard of care and where there's not even a well-established, well-agreed, widely recommended antibacterial standard of care. It may be that standard of care comes down to antiseptic use or other types of things like that. So it may not necessarily be add-on to antibacterial agents or systemic antibacterial agents in all cases. We did want to warn people that failing the primary endpoint in a pivotal trial, but showing a benefit based on one or more secondary endpoints, we would see that as a point in time at which further discussions may be triggered on potentially acceptable primary endpoints in new trials, assuming, of course, that the sponsor still wants to go forward. Switching from a primary endpoint to a successful secondary endpoint and asking us to approve based on that is not likely to be well received. But it's probably worth saying that at this point. For monotherapy, indeed, I agree with what's been said. It may well be that a fairly standard clinical non-inferiority type approach is going to be possible in line with existing guidance, or indeed, depending on the infection to be treated, it may be that we already require a superiority design versus placebo. And so any applicable CHMP guidance should be followed depending on what the target types of infection are. The primary endpoint, indeed, I mean, at this point in time, I think all I can say is that we're looking for a benefit based on clinical cure, on eradication, on a composite of cure and eradication, on a reduction in recurrence rate, and or a reduction in mortality rate. All of those would, I think, at this point in time be considered acceptable. The clinical relevance to the patient of alternative endpoints may be difficult to justify. And I would sort of say that, remember that when we approved the neuraminidase inhibitors for treating influenza, shortening the time to recovery was accepted as the primary endpoint, but there were all sorts of problems associated with that. The HTAs were never really happy about it. And as I think it was David pointed out, here it's not just the regulators that is the hurdle, it's also then getting the HTAs to look at whatever the benefit was and the primary endpoint on which that benefit was shown, and persuading the HTAs that this product has a major contribution to public health. At the moment, endpoints such as reduced duration of standard treatment, reduced number of days on ventilation, effects on selection of resistance, and various other things, all of these are definitely of interest, but we have not yet accepted any of them as primary endpoints. So again, it's worth at least pointing that out. So as I've said, the EMA guidance doesn't specifically address the types of products that we're discussing today, but for the most part, it's highly applicable to them. In the EU, we've had limited requests for scientific advice for products like this so far. Some of them have already failed. Beslituximab was already approved, but altogether, our experience is still relatively limited. I think we have made it clear to sufficient numbers of companies that I can say that the combination of in vitro or in vivo non-clinical data and or clinical data could support proof of concept for the intended clinical use in these settings. As I've said, standard PK/PD approaches may not work here, and so we may be resorting to clinical dose-finding studies, which would need careful discussion with us. There still has to be a clinical benefit at the end of the day, and discussion with us at frequent intervals is essential, starting as early as you want, and particularly to discuss with us the primary endpoint on which a clinical benefit is eventually to be shown. Thank you.
Okay, thank you very much, Mair. And our next speaker is Ian Friedland. Let's see if I can get to your first slide, Ian. Okay, well, Ian is currently a clinical consultant who's been on the front lines and remains on the front lines of clinical development of antibacterials, but including some of these non-traditional approaches. So I'm going to let Ian take it away.
Hello, everyone. I'm going to reiterate a lot of the points that we made by my colleagues. I think, as you've already heard, what we're talking about here encompasses a very broad range of agents with a very broad range of mechanisms of action. And although we're discussing this under one topic, the considerations here are very broad ranging. And it's likely that these different therapeutic classes are actually will have very different development pathways, and maybe eventually will each require their own separate guidance. There is very little experience with developing these kinds of products and very limited NDA, BLA experience. So a lot of what we're talking about here is theoretical. And if one looks at the pathways for traditional indications, like skin infections, UTIs, one thinks over how many decades those guidances have been developed. It's clear that a lot of discussion and adaptation is going to be required before these products are going to be successfully developed. So we talk about non-traditional therapies, but we're talking here more about the development pathways, which are non-traditional, than the therapies themselves. And the pathway one will need to choose will depend on the therapy's mechanism of action. We've spoken a lot about whether they stand alone or add-on adjunctive. And clearly, if it's a standalone therapy, it's very likely it can follow a traditional pathway. So I'm going to focus mostly on adjunctive therapies. These can be preventative or therapeutic. I'm not going to talk very much about preventative therapies. They could be antibacterials, which could include antitoxins, or non-bacterial targets, such as immune modulators. And they could impact both the efficacy or the safety, or one or the other. Now, the novelty, in other words, the degree of prior experience of the therapeutic agent, will determine the complexity and challenges of the development pathway. So the more you can follow a tested pathway, the less the complexity of that development pathway. If the mechanism of action is similar to traditional antibiotics, in other words, they're directly bactericidal, it's very likely you can follow a traditional pathway. However, the adjunctive therapies, where you're adding the therapy to standard of care, is going to be much more challenging than a standalone therapy. And the reason for this is that not only the clinical development may be novel and innovative, but often the non-clinical development has to be different from traditional pathways. So the first step, so you've discovered this very interesting molecule in your laboratory. And the first question to address is this broadly antibacterial, so it can be developed as a standalone, i.e. as a replacement for standard of care therapies, or are we going to add this to standard of care therapy? And that's a very big decision to make, because as all of us have mentioned, if the therapy can be a standalone, it's likely that you can follow traditional non-inferiority trial design. I think the next step, we see a lot of very interesting molecules that are very interesting in vitro or in animal effects. But one really has to seriously address what is the reason to believe this therapy is going to improve current antibiotic therapy. Because the reality is that antibiotic therapy is highly effective in most cases. And if one thinks about the traditional indications, skin infections, community-acquired pneumonia, urinary tract infection, intra-ab infection, antibiotic therapy is very effective in these indications. The only common indications thought, which is hospital-acquired pneumonia, ventilator-associated pneumonia, is the only one indication I can think of off the top of my head where antibiotic therapy is less effective and where there may be an opportunity to demonstrate superiority. I think one really has to look at, once one's decided what indications one might be pursuing, is to examine why is antibiotic failing in these cases, and will your product address directly those reasons? Some of the reasons may be just general reasons, and some may be related to resistance to standard of care. And it's important to determine this because each of these will drive different development pathways. I think for most adjunctive therapies, a broad advantage that can be used in all types of infection is very unlikely. There's definitely a population of patients who are going to respond well to antibiotic therapy. You can't really improve the outcomes. And then there's a population who have irreversible organ damage who you probably can't rescue. And therefore, one needs to try and find the sweet spot where an adjunctive therapy can improve antibiotic therapy. What this means, of course, is that one is narrowing down the unmet need population. And this clearly will have an impact on the commercial opportunity. So the things to consider in the buckets are, number one, for a non-traditional pathway, is what's the non-clinical development? What are the animal models of efficacy you might need to do, PK, PD, dose justification, susceptibility testing, and then the early clinical development, as Mair mentioned, proof of concept may be necessary. This has not been commonly needed in traditional pathways. And dose ranging in phase 1 or phase 2 may be needed. And again, this has not been very common recently with traditional pathways. And then in late clinical, as you've heard, the two most important things to decide on are the indications, the primary and secondary endpoints. Now, the non-clinical development is very important, particularly if one is planning on following one of the streamlined pathways with limited clinical data. This means that the non-clinical development becomes possibly even more critical than usual. And this is especially true if it's a new mechanism of action. One may be able to follow the traditional animal models, such as mouse thigh model, mouse pneumonia, but it may be necessary if one is seeking new or different indications that one may need to explore a new animal model or adapt an existing animal model. And this can be problematic in that there's going to be less experience in extrapolating those animal models to humans, especially regarding the dose estimation. Another question is, can one follow the traditional PD targets, such as time above MIC, AUC over MIC? Are these going to be relevant, or will one need a new paradigm? And if one thinks about how long it's taken us decades to figure out the best way of interpreting traditional PD targets, you can imagine that if you're going to come up with a new PD target, knowing the clinical relevance of this is going to be challenging. And then just a brief word about susceptibility testing. Can one follow the traditional susceptibility testing, dilution testing, disc testing? Because clearly if one has to do some kind of non-traditional susceptibility testing, that's going to complicate the development and possibly even more importantly, the commercialization, because as you can imagine, if clinicians can't use the normal mechanisms they have for susceptibility testing, that clearly is going to impede uptake of new therapy. For clinical development, it's clearly, whatever you do, best to follow the traditional indications, the ones that we know well. If you can do those, your pathway is going to be much easier. If you have to seek a new indication that hasn't been done for a while or has never been done before, and it involves, or if it's even a traditional indication but you're seeking new endpoints, this definitely adds substantial challenges, both for regulators and for academia. I think single gram positive pathogen studies are clearly doable if you have an anti-staph, skin infections, a good indication, strep pneumo, community-acquired pneumonia. I think those are definitely doable. But whether single gram negative pathogen studies are doable, feasible, I think still has to be seen. I'm not aware that there's been recent approvals of single pathogens, particularly the non-fermenters, Pseudomonas, Acinetobacter. May be possible to do Enterobacteriaceae studies and even E. coli studies, but I think the non-fermenters are going to be challenging to get approval just for those non-fermenters. And it's also true, as has been pointed out, if you're dealing with a new drug class, there may be new safety issues, and one may need a new, larger safety database. Not necessarily larger in terms of numbers, but one may need to explore special populations. So that's a thought bubble, not a dark cloud. Translating the clinical benefit of a new molecule when you've had interesting non-clinical findings is not always possible. It may look very interesting in vitro, it may look very interesting in animals, but saying like, how is that going to provide a clinical benefit and how are we going to study that can be challenging. I think of necessity, regulatory authorities are conservative, and the more novelty you introduce into the pathway, the greater the risk of getting through the regulatory hurdles and getting approval from academia and HTAs at a later time point. It's also very likely, and this is a point I feel fairly strongly about, in that these non-traditional adjunctive therapies are less likely to be identified if we follow very traditional endpoints of test of cure on day 30, mortality. And I do think we need to look for alternative endpoints to explore the potential benefits of these therapies. This will require some discussion. This will require demonstration of the clinical value of these non-traditional endpoints. But I really think this is going to be needed to explore some of these new therapies. I think the secret for many non-traditional adjunctive therapies is going to be to identify the population most likely to benefit and exactly how to evaluate this population. Of course, once one starts restricting the unmet need population, I think one then now needs to balance the the ease of the development pathway with the commercial opportunity. And again, how does one translate this more restricted benefit into commercial value? I do think that there are a lot of very exciting non-traditional therapies out there that are definitely worth developing, and I hope we can figure out ways to best evaluate them. Thank you.
Okay, great. Thank you very much, Ian. We're going to move into the question session now. So for those of you who are online, yes, please submit your questions via the questions window, and we'll try and get to as many as we can during this the question session. So I actually have a couple of questions I thought I'd start with while I'm waiting for the audience. So the first one is to Ian, since you just talked about this. So basically, I'm trying to get to this idea of how to demonstrate value. So you have an inhibitor, say, of quorum sensing and Pseudomonas. And you show in animal models that when you combine this quorum sensing inhibitor with meropenem, you essentially take, you can now use meropenem to treat organisms, Pseudomonas, with higher meropenem MIC. So you can shift the target attainment in your animal model from 75 or 80% with meropenem alone to 95% with a fixed dose combination with a quorum sensing inhibitor. Given the paradigms available for development, what are the ways that people can show value for this product clinically in a realistic sense?
Thanks, David. So this is an interesting question, and one that has been faced in similar situations previously. I think this exact scenario that you're describing here sort of boxes you into a one kind of development pathway. I think what you're talking about here is something that can be demonstrated to improve the in vitro potency of an agent, and you've chosen meropenem here, but theoretically one could have chosen some other agent. And this looks very similar to a beta-lactam, beta-lactamase inhibitor, in which the BLI improves the potency of the active agent. And this kind of situation one would develop, I think, very similar to a BLI program, and in particular meropenem is approved for treatment of Pseudomonas. One could follow a 505(b)(2) program here and not necessarily conduct the pathway in the unmet need population. I think you would have to demonstrate the potential value of this, possibly in vitro or maybe with a small clinical study. I think what would be more interesting is if you had a product that broadly improves antibiotic therapy, that is an immune modulator, let's say, for example, and you believe could improve outcomes, and that would have to be an adjunctive therapy added to standard of care, and then that would have to be a superiority trial showing some improvement in an important clinical endpoint.
Yeah, okay, so this quorum sensing inhibitor obviously would not have antibacterial activity itself, and you would not see synergy in vitro. You would only see it when you actually went to your animal models, where suddenly you're able to treat higher MIC organisms successfully in your animal model with standard dosing. So it depends on the in vivo situation. Theoretically, the quorum sensing inhibitor could be combined with a number of any other antibiotic, and theoretically it would have a similar sort of effect. So I guess the question is, do you do superiority trials with versus standard of care, or can you do a non-inferiority trial? And if you do the latter, can you still show value?
Yeah, to do a non-inferiority trial in this setting, and again, because meropenem is an approved agent and you're adding this to meropenem, to me it could follow a 505(b)(2. I think Sumathi can weigh in here. But to do a non-inferiority trial and to assume that there is some benefit from the combination, you would have to be pretty convincing that the non-clinical data is adding a true benefit. Because if you have to demonstrate this clinically, just in Pseudomonas, I think that would be very challenging. So I think if you could figure out a pathway that involves non-inferiority, just showing that your product, is similar to existing standard of care, but the advantages can be demonstrated in vitro or in animal models, and that's convincing, that would be a better pathway than having to seek clinical superiority.
And do you think you'd be able to convince the end users, physicians, patients, payers, or even investors with that sort of approach?
And I think you've hit on something. This is tough, as you know. Anything that's new, does require, a fair amount of convincing, both to academia, and to people who are going to help you develop things commercially.
Okay, great, thanks. Do Mair or Sumathi, do you have anything that you wanted to add here? Okay.
Nothing from me, thanks.
Okay, so I'll go on to the next one here. This is kind of a question for everybody, but I think one of the issues that's come up in thinking about trials in general, both non-inferiority or superiority trials, is I've seen a number of companies thinking about adjunctive therapy to colistin or polymyxin containing regimes for resistant gram-negatives. And yet there are a number of studies now that show that other drugs, plazomicin, ceftazidime, avibactam, and meropenem, vaborbactam, especially the latter two, are all superior to colistin-containing regimens for treatment of these resistant infections. But on the other hand, these new drugs are often not on hospital formularies. They're frequently not available in certain parts of the world. So for everybody, is colistin polymyxin still an acceptable comparator? Actually, whether you're talking about non-inferiority or whether you're say an acceptable comparator regardless of superiority or non-inferiority when it's included as part of a standard of care or best available therapy regime. What do you all think about that? Maybe we could start with Sumathi.
Yeah, sure. Thanks, David. It's a very interesting question, and certainly one that we've heard discussed at different fora. I think it raises some important questions, and first and foremost, yes, I would agree that some of the recent data suggests that the newer antibacterial drugs may have a better outcome. I mean, at least there's a trend. I don't know if any one of the studies has conclusively shown us that, but I think it's fair to say that there seems to be a trend. But I think one of the biggest difficulties with superiority trials in the antibacterial space is I think, highlighted by this question. It is very difficult to demonstrate superiority over existing therapy. So, there seemed to have been a small window when one could potentially demonstrate superiority over maybe colistin-based regimens, but as new therapies become available, that might be harder and harder to do. But really, from a practical standpoint, when we've seen proposals where a colistin-based regimen has been proposed as the best available therapy, and we have questioned why the other therapies are not being considered, I think the most common response we have received is that they are just not available. And especially for studies done across geographic regions, I think there's a fair degree of variability in the availability of some of these therapies. And so it all, I think, comes down to if there is clinical acceptability or not. So it's, that's kind of what would be my comments.
Great, thanks. Mair, do you have anything that you wanted to add to that? What do you think about this?
Well, first of all, none of the studies were designed to conclusively show statistically superior effect to colistin-containing regimens. So I agree that there are trends, but we have to also be very careful about making statements that they are superior. I think here we're really talking more about some traditional pathways. And indeed, we have started to encourage companies where possible to think about using Cazavi or Weber-Mair as the comparator in certain non-inferiority trials, simply because that means that they can open up enrollment to people who are known or suspected to be infected with CREs without having to resort to randomizing people to the rather more toxic colistin polymyxin alternatives. So we are moving in that direction, encouraging people, but I think we also are not demanding that, at least not yet.
Okay, great, thanks. Ian, do you have a thought on this?
Yes, a couple. So we're talking about here are fairly traditional pathways. I would not undertake, given my experience with plasmamycin, a CRE-specific trial and try and demonstrate superiority. I don't believe that's feasible. I think colistin could be okay in a superiority trial. I definitely don't think it's suitable for a non-inferiority trial. I think I'd have a lot of problems with that and would rather use one of the new agents. It does raise a very interesting question: is at what time point do new therapies become the new standard of care that one can use in a clinical trial? And obviously there's no right or wrong answer there. But obviously it does take some clinical experience, it does take some acceptance from the ID community that this has now become the standard of care, I think, before you could realistically put that in a clinical trial.
Yeah, okay, those are exactly the kind of points I wanted to get at, so that's very helpful. We have a few questions from the audience. I'm going to try and pick here. So one of them is for an immunomodulatory therapy, is there some species selection that's more relevant to look at PK/PD or even just pharmacology? Or for efficacy models to look at these immunomodulatory agents? Anyone can chime in on that one. How about Sumathi? You've thought about this. What are there species you think are more relevant for an immunomodulatory therapy?
Yeah, unfortunately, I don't think I have an answer to that question. I think in general, our advice has been that people come and talk to us and explain to us why they think the specific model they have chosen is relevant to their product. I mean, otherwise it's really hard for me in a vacuum to say this model is better than the other, and I don't think I have the expertise to provide that advice. But I think What's more important is to have a good reason for why you've chosen your specific model and what is the assessment you're going to make in that model.
Okay. Anyone else have anything to add there?
Well, the choice of model is also, in some fields of anti-infective development anyway, driven greatly by the type of infection or the specific pathogen that you are trying to show an additional benefit for. I think it's really complex and it has to be decided case by case, but the model is not just driven by being able to get as near as possible to mimicking what happens in the human immune system. It's also driven to some extent by what sort of model is useful for the type of infection or the specific pathogen that you're after.
Okay, thanks. So another question that's come up has to do with protein-based therapies, for example, license, phage product license. What non-clinical studies do the regulatory agencies normally consider are sufficient to look at immunogenicity for these sorts of products? I know there are guidance documents that talk about this, but Mair, could you start with that one?
The question was about non-clinical species?
Yeah, so non-clinical species, what non-clinical studies are normally sufficient or required to assess immunogenicity for protein-based therapies, such as, for example, a phage lysin?
I think that the choice of non-clinical species, I mean, it's covered in the guidance, but again, it's got to be something that's relevant to the type of product. But the critical thing is to look for anti-drug antibodies in humans and to assess whether they're actually doing anything or not, because very commonly these products do elicit anti-drug antibodies, but they don't impact in any way on the safety or efficacy of the product. So we would expect that, I mean, we have standard guidance on this and it would have to be followed, but I would put much more weight anyway, ultimately, on human ADAs rather than non-clinical studies.
Yeah, okay. Sumathi, do you have anything to add there?
No, I think in general, we advise people to look at the ICH S6. I think that's pretty clear about what kinds of studies. And again, a lot really depends on the product. And these are general guidelines. And if you want to do something different, and I think it's as long as you explain to us, why with your particular product, you don't need to do 2 species. I think ICH S6 recommends 2 species. Why is 1 enough? And why that particular model is relevant, I think that's why I think all of us had emphasized in our presentation, it's very important to have a dialogue and provide a rationale for the approach one is taking for your specific product.
Okay, I have an early question here, which has to do with an antimicrobial agent that's a combination of a peptide and an oligonucleotide. And they're asking, is this considered a traditional or non-traditional category? Basically, I mean, I think I can try and say that it depends on how it's going to be used, really. I guess the other question, part of the question would be, how would the CMC of such a molecule be handled? And for that, I have no idea. Do the regulators have a thought on that?
I mean, again, I think I provided a link for what we consider biologics and what we don't consider, and in that there's a lot of information about peptides and the size and the number of amino acids, etc. So for us, I think all that's different is the chemistry groups that support a biologic product versus a non-biologic product is different, and obviously the requirements are different. The guidances are very specific, whether you fall into the bucket of biologic or non-biologic, but the general principles, I'm not sure if there's a big difference. And I think all of those specifics, when we see the product, I think we make a determination and triage it to the appropriate group.
Okay. Mair, do you have anything to add to that?
No, except to say that in the EU, it's only the quality assessors that would split between the biologicals and the non-biologicals. And that the non-clinical and clinical people who look at them will essentially be the same.
Okay. Now I have one question here that I don't understand, but I think theoretically you guys will understand it. It says CBER in the US has interact meetings, targeted engagement for regulatory advice on CBER products, which has a very limited scope. Any plans for CEDAR or EMA to provide early discussions prior to the pre-IND meeting? Actually, I mean, my experience with FDA is you can just call them and meet, so I don't know what that means. But can you guys deal with that?
Mair, do you want me to go first, or I know you had started saying something?
Well, I was just going to say, sorry, David, I thought you'd finished, but I mean, we do have an ITF system as well in the EU, which provides a pathway for which, you know, products that are away before human use can come along and just discuss in principle what they think their product can do and what sort of program we might be looking at some time in the future. That's even outside of scientific advice. It's just called an ITF type of meeting.
Okay, well, that sounds like an answer. How about Sumathi, am I right that you can come in anytime for a so-called pre-IND meeting, right?
Quite right. I don't have any specific acronym, but if you have a question, come talk to us. And really, we are very, very flexible. We don't dictate that you can only come to us at a certain stage. If you're very early in development, it might be like a pre-IND, but that's okay. We'll still meet, provide feedback.
Okay, I'm going through these questions here, and I have to say that some of them I don't understand. So, okay, well, I have one that's a bacteriophage question, but I think that maybe this is the wrong group to a certain extent. Wow. I'm sorry, I'm just reading these questions. Yeah, so I have one question, which again, I'll try and handle a little bit. I mean, basically, the question is about the idea that it's one thing to develop a drug for a regulatory approval using clinical indications where you have clear guidance and you know how you're going to get it approved, et cetera. But the way physicians use the drug is quite different often, which can be both a good thing and sometimes maybe not such a good thing, but it's the way it is. But I don't see a question here. So if the person who submitted this can give me a question, I don't know, but it is true that people get approved for indications is one thing, and then the way physicians use the drug is frequently or can be something else.
So Dave, that actually raises a very interesting situation in that it clearly demonstrates the flexibility of the regulatory authorities in that you can pursue an indication and get approval for a drug because you can demonstrate efficacy and safety. But the indication that you have actually pursued is not the biggest unmet need. So for example, these CRE active drugs were approved largely or certainly partly based on showing efficacy and safety in urinary tract infection, which is not the biggest unmet need. And that does raise the slight problem in that you've done the trials, you've got the data in that indication, that's what your label specifies, but the unmet need is actually slightly different. And how do you exactly go about commercializing that product when the unmet need is not specifically what your label says?
I think the question, I do have a clarification from the person who asked this question, and it is basically, can the regulators do something to address this? Like, could there be streamlined development for the unmet need indications to allow them to allow the drugs to be labeled for the patients who have the unmet need? And the answer is that that guidance is already available, and yes, it can, but it's a lot harder. And so most people, if they're going to do it, will do it later, as opposed to doing it up front if they don't have to, such as for a standard antibiotic that can be approved for a standard indication such as urinary tract infection and intra-abdominal infection. So I think the pathway is at least theoretically already available from the regulatory authorities.
And David, in the EU, of course, we also have the possibility of a pathogen-specific indication. And for some of these novel therapies, they are being directed at very specific species or genera rather than against types of infection. So I think in the EU, we could end up handling some of these, provided that we are convinced they do potentially address an unmet need, the way we would handle and indicate standard antibacterials that we think can address an unmet need.
Yeah, I think the FDA has a similar pathway. Actually, they have the unmet need pathway. They also have LPAD available. Sumathi, would you agree that those are kind of, I mean, they're not the same as the EU pathways, but they're similar?
Right, and I think our guidance is, as you've mentioned, clearly articulate what might be options in terms of development programs. I mean, we've certainly heard some concerns raised about, why are products being just developed for CUTI, but I think the practical difficulties of doing a trial just in, say, a particular phenotype is very challenging. And I think Ian's mentioned that we've all learned from some of the recent clinical trials done. So if that's the pathway someone wants to pursue, I mean, we're happy to work with them, but I think there needs to be an understanding that doing those trials is going to be very, very challenging. And being able to demonstrate superiority might be particularly difficult. So at least having the drug evaluated in an indication where one can make a reasonable assessment of the efficacy and safety of the product is at least one step in getting the product available for patients, and then continuing to build upon that and then pursuing the more difficult indications, I think seems like a very reasonable approach.
Yeah, Mair, I'm aware of 2 companies that are pursuing pathogen-specific indications in phase 3 at this point. Since your guidance, since the EMA guidance has come out in January, are you seeing a lot of people coming forward with questions about how to actually do this?
Well, what we're asking for hasn't really changed. And the point is that we will translate a clinical program into a pathogen-specific indication without restricting it by infection site in certain specific circumstances. So instead, I mean, if a trial is done in CUTI, but we also have a very convincing package of PK and PK/PD, and we see that the drug addresses an unmet need, we would give a pathogen-specific indication. And I think that is the difference versus the US.
Okay.
And therefore, given that we would do that for a standard antibacterial, I don't see why we would not do it for a non-standard type of product, provided, and this is the critical issue, we are absolutely convinced that it would address an unmet need.
Yeah. Okay. That's clear. Thank you very much. Well, I'm kind of running out of questions here, and we're approaching the end anyway. Do any of you, Ian or Sumathi or Mair, have any last words you'd like to provide for the listeners? Okay, well, I'm going to provide it for you. The bottom line is this is a risky area. and it's going to require a lot of consultation, not just with the regulatory authorities, but with the potential base of users, clinicians, and payers, and sometimes, in the case of small companies, investors, to get a good understanding of the risks involved and how you're going to be able to deal with those risks in the context of some of these non-traditional therapies. And with that, I'd like to thank everybody. I'd like to thank certainly our speakers. I'd like to thank GARD-P for continuing these efforts, and I hope people will find this to be a useful webinar. So thank you very much, everybody. I think we can close out now.
Yeah, thank you.
Thanks, David. So I would also like to thank all the speakers for their time and for sharing their really invaluable expertise with us. And a special thank you to you, David, for organizing these sessions. I would also now like to take the opportunity to announce our next webinar. On the 9th of April, we will have Karl Balliber from the Merck Research Laboratories on the line to talk about mining of chemical libraries for new antibacterials. You can already register for this webinar on Revive, and we will soon also announce our May and June webinars. With this, I would like to thank everybody for joining today and for contributing to the discussion. I really hope that the webinar was interesting and useful for you, and that you will join us again in the future. I would appreciate to receive your feedback in the survey, which will pop up right after this webinar, or also per email. Thank you, everybody, and goodbye.
Thank you. Bye, everybody.